Finished products should be examined for microbial contamination to ensure that all phases of the production and sanitation systems are functioning properly. We'll be testing for the following bacteria: coliforms, yeast and mold, heterotrophic plate count. To set up your pump and manifold, cut 3 sections of tubing, one 8-inch and two 12-inch. Then securely attach the tubing to the pump and manifold. Disinfect the work area with either 70% isopropyl alcohol or an alternative disinfecting agent and allow the work area to air dry. While you wait, you should put on both protective eyewear and gloves. Be sure that the valve for each monitor is in the closed position. Take the microbiological monitors out of the box and position them on either the vacuum manifold or a filtering flask using the adapter provided. Each monitor comes with a removable lid, a measured filter funnel, and a membrane. For this demonstration, we will be using 2 white 0.2 membranes and 1 black 0.8 membrane monitors. The reason for choosing the 0.8 membrane is to obtain a more accurate reading when testing for yeast and mold. The 0.8 membrane will allow other smaller bacteria to pass through. From the water sample collected, pour 100 milliliters into each monitor, then replace the lids. Turn on the vacuum pump and then turn each valve to the open position. This will allow the vacuum to pull the product samples through the membranes. Turn off the vacuum after the entire product sample has passed through each membrane. Break the vacuum of each monitor. Then, once again, position the monitors securely back onto the vacuum manifold or the filtering flask. Now, move each valve to the closed position. Lift each monitor lid and pour 99 milliliters of sterile rinse water into each funnel. This will ensure that any organisms of bacteria that may remain on the sides of the funnel will be rinsed and displaced onto the membrane. Turn on the vacuum pump and turn each valve to the open position. After all of the rinse dilution is passed through the membrane, turn off the vacuum. At this point, it's very important to break the vacuum again on each monitor and close each valve before you move on to the next step of applying ampuled media. The entire contents in each dose applicator of media must be used to achieve accurate test results. Slowly squeeze the MI coliform media onto the top surface of the membrane. Be sure not to splatter the media onto the sidewalls of the funnel. It may be necessary to rock the monitor carefully back and forth to completely coat the entire surface of the membrane. Now, slowly squeeze the yeast and mold media onto the top surface of the membrane. Again, be sure to completely coat the entire surface of the membrane. And finally, we'll move on to the heterotrophic plate count media. In order to easily dispense all HPC media, the ampule must first be placed in warm water. Then, slowly squeeze the HPC media onto the top surface of the membrane, completely coating the entire surface of the membrane. Turn on the vacuum pump. Open each valve, leaving the valves open for only 1 or 2 seconds, just enough time to allow the media to pass through the membrane. Then turn off the vacuum. Remove each monitor from the base. Now, without twisting, remove the funnel portion and discard. Then place the lid onto the base to form a petri dish. Securely place a plug on the bottom port. The final step in this process is recording the data code of the sample that you have just tested. Simply write this information on the cover of the petri dish. To prevent condensation from dripping onto the membrane, turn each dish upside down and place in the proper incubator for the appropriate amount of time. Incubate each sample according to the table found in your manual.